So I'm coming a bit more from the peptide and protein chemistry side, and hope to give you a bit of a perspective of some of the things we do in terms of protein and peptide chemistry, some of the problems we currently face, and some that we've partly overcome. So you've had plenty of time to read my title now. So I'm sure anyone who has worked with proteins, or even if you haven't, will be familiar with the concept that DNA, RNA, protein, and often the story ends there. But what we sometimes forget is that that's not really the end of the story, and that almost all eukaryotic proteins at some point in their life cycle are post-translationally modified. And these post-translational modifications can vary from very small groups like a small methyl group, a phosphate group, to slightly bigger things like lipid chains and sugars, to some really fancy complex sugars and even other proteins like ubiquitin. Those of you who've worked with the tutorials might recognize it. So these proteins and these post-translational modifications are some of the ways that a cell regulates the activity of a protein without having to go all the way back to the beginning and make a whole new protein to change the function or perhaps to respond to certain stimuli in the environment. And these post-translational modifications and their effects on protein structure and function are what we're trying to investigate in my group from a chemistry and from a structural biology perspective. So those of you who've expressed proteins in bacteria will know that usually you get the unmodified version because most of our typical expression systems, especially in bacteria, E. coli, are not very good at installing eukaryotic post-translational modifications. So if we want to understand what a particular modification is doing in a protein, we need to access that protein with that specific modification in exactly the place we want it, and ideally only that place. So there's a few ways we can do this. Nature, of course, often uses enzymes. We can also use these enzymes, but for almost most of the post-translational modifications, either the enzyme is not known, or it's not accessible, or it can be very difficult to get the enzyme to put the modification exactly where you want it. In natural systems, they have many other, say, coenzymes and binding contexts where they might site-specifically insert a modification, but that's very difficult to control in vitro. Genetic code expansion is a very powerful tool for generating modified proteins, where you kind of overwrite or rewrite using a stop codon to insert an unnatural or non-canonical amino acid at a particular position. And this is very powerful in that it happens already in the cell, but it's not usually practical for NMR structural biology purposes because it's not so compatible with isotope labeling. And then you also have to isolate the protein, typically from a complex mixture, and the yields tend to be very, very small. My favorite method, and of course I'm slightly biased, is using protein synthesis and semi-synthesis. We can also use chemical ligations. Why is this not moving forward? Chemical reactions that react site-specifically, for example, with specific side chains. Often we use cysteines and lysines and things. Those tend to generate modifications that are not exactly identical to the biological modification, but can still be very useful. But the rest of my talk, I'll focus mostly on protein synthesis and semi-synthesis, because it's, I believe, one of the best ways to very site-specifically install a modification or non-canonical amino acid, and I think also has quite some power for looking at structure. So for those who perhaps not so familiar with the chemistry side of peptides, just a very brief overview. Typically in the lab, we do this either manually. We still do quite a lot of manual synthesis, especially for tricky peptides and expensive building blocks. So automated synthesizers, there's many of them around. They can be not quite so easy as some students believe. You just type in your sequence and press go. Unfortunately, it's not that easy. but can make life a little bit faster. So we synthesize the peptides on a solid phase resin, build them up amino acid by amino acid through a series of coupling and deprotection cycles. And the cyclic synthesis means that at any point along the peptide, we can insert basically whatever we like. So this could be an unnatural amino acid, could be a D-amino acid, could be an isotope labeled amino acid, very site-specifically, atom-specific labeling, or other handles for other reactions, azides and tetrazenes, fluorescent tags, biotin, basically anything that the organic chemists can make. So this is a very powerful tool for making site-specifically modified peptides and proteins. Those of you who might have done some peptide synthesis realize that there's something of a size limit. And because we have this iterative process, once you get beyond about 40, 50 amino acids, usually the quality of your synthesis goes downhill very fast. And most proteins are much bigger than this. So here we come to native chemical ligation, which is there's several tools. This is probably the most widely used and most versatile for joining segments of proteins, peptides, together to assemble a whole protein. And these segments can either be all synthetic or partly synthetic and partly recombinant. The reaction works like this. You need a C-terminal thioester on one segment. You need an N-terminal cysteine on the other segment. They undergo trans-thioesterification to form this branched thioester intermediate, which then undergoes a spontaneous S to N acyl shift to generate the native amide bond. There's now many variants of this, and it's been extended much more to not really require a cysteine at the ligation site. We often use a cysteine and then desulfurize it to an alanine. So that gives us a lot more choice of our ligation sites. There's a lot more alanines in proteins than cysteines. And the advantage is that really you get your native peptide backbone. It's fully amide backbone, you can't tell where the join was. And we can then stitch together lots of bits and pieces of peptides and proteins. So I've given a bit of introduction into kind of how we approach a protein, more from the chemistry side of how we're going to make it, how we're going to make it with a very specific modification. But now if we want to understand what this modification does to its structure, its dynamics, we run into a little bit of a problem. Because protein NMR is very well established and typically uses these three very common isotopes, which are great for proteins. Obviously, the protons come for free. The carbons and nitrogens, if you want to synthesize a peptide isotope labeled as you would for a recombinantly expressed protein, this becomes prohibitively expensive. Some of the building blocks are not available. Some are available, but you, at least as an academic lab, we certainly can't afford to make a whole peptide out of isotope labeled amino acids. Inserting specific ones along the sequence at a particular point that we're interested in is certainly feasible, but the whole thing, certainly not. So then we have to take a step back from the traditional kind of carbon-nitrogen labeled triple resonance spectra and say, well, what can we do with the protons that we get for free? Or can we make enough sample that we can use the natural isotope abundance? And this is just to remind you that there's unfortunately not much of them. So these are some of the ways we get around this, and I'll talk briefly about the approach we use for each of these, followed by a very short case study of a research project that we've done to illustrate that. So one way we could get around this problem, is we could just stick with small peptides. Then we can make lots of them. Usually they're reasonably soluble enough that we can get enough in the NMR tube to use carbons and nitrogens, also small enough that we don't land up with too much overlap in our proton spectra that we can still assign things. Or we could take our big protein that's modified and say, well, okay, well, let's just forget the rest of the protein. We just take the modified bit and we look at that bit. can also be a valid approach. Of course, you always have to justify why you forget the rest of the protein, and that can be difficult. Or we can turn to segmental labeling. And here we come back to native chemical ligation, because this we, again, kind of get for free. Because if we're already making our protein out of bits and pieces of synthetic and recombinant bits, we can then mix and match labeled and non-labeled segments. So this can be an approach in that we could, for example, synthesize the segment of our protein with the modification, and then ligate it to the rest of the protein expressed usually in E. coli, which we can label using standard fashion. And then we land up with this funny kind of hybrid protein where you wouldn't be able to tell from the backbone that there's no chemical difference, but part of it is labeled, part of it is not labeled. The unfortunate part is that the part that's not labeled is actually the interesting bit that you're trying to look at. So then again, we come to this problem of how are we going to get around that? And with some of our projects, we are looking at interactions between labeled and unlabeled segments. This mouse is doing weird things. I'm quite interested in looking at experiments where we can look at interactions between labeled and unlabeled, filtered and edited experiments. In others, we are trying to switch where we label it so that we can look at different segments of a protein at a time. And this can also be really useful. One example that I haven't included here is the rather large tau protein, 440 something amino acids, where we could then use segmental labeling to avoid signal overlap and really work out which of the segments, especially in these highly repetitive, intrinsically disordered proteins, which is which. Excuse me. So I'll just go back here. So I'll go through these several strategies that we have in looking at peptides and proteins using at their structure and function. But first, a brief overview of some of the typical workflows that we think about for peptides. And I was reminded this morning that, yes, perhaps there's some of these that are now perhaps a bit old-fashioned, and that we could ideally automate and interface a little bit better with each other. And it's been exciting to see already this morning I learned of some new developments where that's got a lot better than when I did these projects before. So I'm also very glad to learn what's going on here. So typically we make our peptide or protein synthetically and or ligate it. Of course, fold it if it's a folded protein. These days we tend to work more with intrinsically disordered proteins, which then brings some other difficulties in terms of signal overlap, but at least it means you don't have to fold them. We then acquire our NMR data. And here, if we are working with natural abundance isotope, a lot of our data is heavily focused on proton-proton, toxi, and nosy data. So that's where we get most of our information from. If you have enough material and a higher resolution enough instrument that you can get some time on, you might be lucky and also get a carbon and nitrogen HSQC, which is incredibly useful, as I'm sure you know. But triple resonance data is not really an option. We then try and assign it as best we can, usually very manually. Usually they're small enough to really pick peaks ourselves and assign them. Some of the automated assignments we find don't work terribly well for fancy peptides with modifications, cross-links, cyclization. There you really have to go manually one by one. Once we've got the assignments, we assemble those with secondary chemical shifts, distance restraints, atomic descriptions and coordinates, and whatever dihedral angles we can get. Go through our rounds of structure calculation and reassignment until we have our ideally nice ensemble of structures. So fairly traditional workflow, but a slightly different perspective in that we're heavily focused on the proton-proton 2D data. So when I started doing this workflow with some modified peptides and proteins, I realized that there were a few gaps. And one of these was in the secondary chemical shifts. Some of you might have worked with Talos or with random coil shifts or even just average chemical shifts of natural amino acids. But many of these were missing for the post-translationally modified amino acids. So I started with a small project before I even got going to assign random coil shifts for post-translationally modified amino acids. And here I made a small set of just short random coil peptides with the modified residue in the middle. This was the set that I worked on. Since then, there's been a few more. This fairly standard post-translational modifications, such as phosphoserine, threonine, tyrosine, some hydroxylations, arginine methylations, some slightly fancier ones, a couple of simple sugars, and used those to assign random coil shifts. And just for example, this was already known before, but now assigned in a hopefully systematic way, the typical downfield shift of a phosphoserine NH peak because of this hydrogen bond that forms between the phosphate group and the backbone amide. So that was more just to recognize the patterns, that random coil shifts of post-translational modifications, so that we can start to include them in structure calculations and not always have to substitute them for the kind of closest amino acid that we can make do with. I also got involved in a very small way in a collaboration with some researchers at the University of Queensland, where we were looking at incorporating non-canonical amino acids into assignment and structure calculations. And this is something I learned more about this morning, a slightly different approach that's now integrated into CCP. I think still compatible with this, and perhaps we can work a little bit more at bringing these two together in talking to each other about how to handle sort of fancy peptides where you might have unusual bridges and connections and non-standard amino acids. So that's one example of just some of the groundwork in figuring out how do we handle these modified peptides and proteins in a standard structure calculation workflow. The other option, now that we've got some tools for handling these, is simply to work with small peptides. And these are actually quite fun to work with. One example that I worked with is the small conotoxin. This is a toxin produced by these cone snails that live on the Great Barrier Reef in Australia. And they produce these toxins that are very highly modified. This is just one example. Has two disulfide bonds, a C-terminal amide, and a tyrosine sulfation. So if you consider the length of it, 17 residues, and already 1, 2, 3, 4 modifications, this is a fairly highly modified peptide. Would be difficult to produce recombinantly, so we made it synthetically. And then we could make all the different variants with and without sulfation, with and without amidation, to see what these modifications are doing in terms of the structure and function of the peptide. So we first of all tested their activities on, this one is active at a nicotinamyl acetylcholine binding receptor. And we could see that as we add in the modifications, the tyrosine sulfation and the C-terminal amidation, we get an increase in the activity. We also get increased binding to a acetylcholine binding protein, which is the mollusk equivalent of this receptor. So these modifications are obviously doing something to the activity of the protein, but are they doing something to the structure? So then we took our synthetic peptides, did some NMR structures of them. I won't go into the data, but just briefly to say, the structures are not particularly different. They're highly constrained by these disulfide bonds, but we noticed some small differences in the modified variants. For example, a bit more definition in these turns at the end, and interactions between the C-terminal amidation and the backbone here, as well as the tyrosine sulfation, which is negatively charged, and the positively charged N-terminus. So I show this just as an example that these small conotoxins, small peptides, can actually be great model systems for investigating these post-translational modifications. because they're small enough and easy enough to synthesize, and we can do relatively easily get NMR data on them at natural isotope abundance. And we now have tools to handle the post-translational modifications and include them in structure calculations. But as I said, not so many proteins are nice and small and convenient to handle. Often we're interested in questions of bigger proteins or segments of bigger proteins. And this is an example of what a project that I've been working on that is based on the HMGN1 protein. Just very briefly to introduce it, it's an intrinsically disordered protein. It's not big as proteins go, it's around about 100 residues. And I thought, okay, that's a nice size for NMR. It was one of the reasons I picked it. It's thought to be involved in chromatin folding in the compaction of DNA and to interact with the acidic patch of that the histones form on the nucleosome. And of course, in my interest, it was highly post-translationally modified. These are some of the modifications that are known in the UniProt database. Of course, they don't all occur at the same time, but if we want to know what they're doing and what effects they have on structure of HMGN1 and its interaction with nucleosomes, then we need to be able to access modified variants of this protein to see what the modifications are doing in, I'd say not really changing the structure because it's intrinsically disordered, but in altering the dynamics and conformations of this dynamic protein. So to start with, we took just a section of HMGN1, the nucleosome binding domain, which is the central part here, and looked at the 2 phosphorylations on these 2 serine residues here. And this was about 30 residues long, so reasonably accessible through solid phase synthesis, and we could make all the variants, one phosphorylation, the other phosphorylation, or both. We also experimented, this was very recently after the AlphaFold 3 came out, and we thought we'd just have a go and see. Now you can include a few limited post-translational modifications in structure predictions with AlphaFold 3, and we were kind of curious to see what it did. So I say this with a bit of a, a caveat, in that we don't really believe these structures that we see because we know AlphaFold 3 doesn't handle peptides well, it doesn't handle post-translationally modified things well, and it doesn't handle intrinsically disordered things well. So with all those caveats, we were just kind of curious to see what had popped out. And we're interested to see that, yes, it predicts a little bit of helicity. I won't overinterpret this because, as I say, there's not a lot to interpret from it. But we were interested to see slightly different amounts of helicity in comparing the few variants. And we then tried to look at the experimental data to see, is there something in this, or is it just completely making up stuff? So we looked at the CD data of all these variants and saw indeed they are intrinsically disordered. There's basically no alpha helicity here. Perhaps some slight variation between the variants, but this could also be very slight concentration differences, which are very hard to optimize with protein small peptides that have no fluorogenic amino acids. We also tried to force it into a helical structure using trifluoroethanol and to see which peptide is easier to force into a helix than others. And here it looks like the unmodified and the phosphorylated variant at phosphoserine 25 are a bit easier to force into a helix than others. And we're trying to understand that with a couple of other examples now as to if you force something into a helix, how easily can you force it into a helix or force it out of a helix? And can we interpret that in terms of partial structure or structural, like, what's the word? Not predetermination, but structural propensity, or is that a bit too much of an artificial system, kind of forcing it into a helix with trifluoroethanol? Then, of course, we put them in the NMR spectrometer and got 2D data. I must say, even for 30 residues, this was, I think, one of the hardest assignments I've done because it's basically all arginines and lysines and prolines, and everything sits on top of each other. So this was really challenging even for 30 amino acids. And our secondary chemical shifts show that there's very little structure. I've blown them up a bit, but the magnitudes are tiny. Perhaps some differences in the C-alpha chemical shifts comparing the variants. And this is, again, something we're trying to see which experimental methods can we use to tease out these kind of subtle differences between structural propensity or different conformations and learn something about those. Here we did get an N15 spectrum quite nicely. And here again, we can see the very typical downfield shift of the phosphoserines and some of the neighboring residues. And I'm trying to work out what that means in terms of, is that altering structural propensity or not? Is it shifting the populations of this intrinsically disordered protein? So that's something I'd be certainly keen to discuss with anyone who would like to, what we can learn from these data, if we can perhaps get more out of them than we are getting out, or perhaps ask better questions so that we can use this tool of being able to really compare variants and get experimental data and see if our predictions are actually following the same trends as what the experimental values are, which in this case they were. So interestingly enough, the AlphaFold predictions, although we don't believe there are really helices there, like we like to see nice clean helices formed there, the trends actually follow all the experimental data as well. So perhaps there is something there that could be telling us something, but that's something we're trying to work out, is how subtle is it and which experimental data are best to use for that. Finally, I'll give a small example, sticking with the HMGN1, of how we used segmental labeling to now look at the full-length protein. If you remember, in the previous study, we looked at just the synthetic variant of the nucleosome binding domain. In another small project, I looked at the phosphorylations in the N-terminus and the C-terminus, and then acetylation at lysine 2. So now looking at the two ends and making the HMGN1 semi-synthetically. So if we do this, first of all, we express the whole protein fairly traditionally using recombinant expression, try to isotope label it so that we can see what the whole protein looks like. So this we could get really nice, clean data. CD again showed that it's completely disordered. The NMR data also showed the same. This we managed to assign despite quite a lot of overlap. You can probably recognize a very classical intrinsically disordered protein with everything having very little dispersion here. And then I turned to semisynthesis to look to make specifically modified variants of HMGN1. So here I looked at the N-terminal segment, made that synthetically with the modifications that I wanted, used native chemical ligation to ligate that to the rest of the protein, which I'd made recombinantly and isotope labeled. So I could, we introduced a cysteine here for the ligation, then desulfurized it to get back the native alanine. And so then we had the full-length native HMGN1 sequence with site-specific modifications, either nothing, the lysine acetylation, the serine phosphorylation, or both, and look and compared these in the NMR spectra to see if these modifications are making any difference to the conformational populations. And here, of course, remember, we're looking at changes in the recombinantly expressed labeled part caused by modifications in the unlabeled synthetic part. So these are fairly long range and very small, but we think significant in terms of specifically the phosphorylation at serine 6, causing some chemical shift perturbations in the nucleosome binding region. And I'm now trying to make differently synthetic, segmentally labeled variants of it to see really which modifications are causing these shifts, and if that means anything in terms of folding and in terms of nucleosome binding. Just as a quick illustration, I also did it the other way around to look at phosphorylations in the C-terminal domain, using a ligation junction here, again with a cysteine, but this time expressing the N-terminal part recombinantly and the C-terminal part synthetically with the phosphorylations, ligating, desulfurizing, to make a few variants with different phosphorylation patterns. Here we didn't see such big shifts in terms of the nucleosome binding domain, which is now really quite far away for this intrinsically disordered protein. But some slight shifts in the nucleosome binding when we looked at electrophoretic mobility shift assays. Of course, we also looked at the synthetic segments by themselves and could see some of the shifts of the phosphoserines and some neighboring residues there. So now looking at bringing those two together, the semi-synthetic and the synthetic parts, and trying to work out what these modifications are doing in terms of interactions with the nucleosome binding domain, and ideally then with the nucleosomes as well. So just to recap, working with synthetic peptides gives us great access to specific modifications, but not great access to isotopes. But there are ways around that. Either we just work with the small peptides, or we can stitch them together into bigger proteins and use segmental labeling to look at bigger proteins and longer range effects. Finally, I'd just like to thank my small team at the Technical University of Vienna, collaborators at the University of Vienna and the University of Queensland. I was grateful for an INEX project where I could get some NMR time in Utrecht with Hugo van Ingen to acquire some of the NMR data that I showed here today. And looking forward to your discussions afterwards. Thank you.